The next speaker is Dr. Atack, who is a senior scientist working in the Department of Neuroscience Drug Discovery at Johnson & Johnson in Belgium. Dr. Atack has been working on aging and Alzheimer research in Newcastle, in Washington, D.C., in Harlow, and in California, and before joining Johnson & Johnson. Please, Dr. Atack.
Thank you. So I'd like to thank Peter and the organisers for the opportunity to be here today and talk about some work I've done in the past on GABAA receptors. We've heard a lot about GABA this morning and this afternoon. I should explain I'm currently employed by Johnson & Johnson, and Johnson & Johnson makes baby powders, it makes medical devices, it also makes drugs. And the drug division is called Janssen, so my badge says Janssen. I'm employed by Johnson & Johnson, but actually I'm going to talk about something that I was working on with my previous employer, Merck. So a little bit confusing, but bear with me. And I'll draw your attention to the press release that was in the registration documents when we got here this morning. Press release about Roche conducting phase 1 studies with a GABA compound for the treatment of Down syndrome, which I think is a terrific achievement. There's a couple of ladies at the back there I'll draw your attention to. They're from Roche, and I wish you all the best of luck with these studies. The kind of molecule that they're studying is very similar to the kind of molecules that we were studying at Merck, and that will be the subject of my presentation today. So in terms of cognition pharmacology, as Mario very eloquently stated previously, we have certain weaknesses in that the predictive value of animal models is questionable, to say the least. So that's a definite weakness. So how can we predict that compounds that we test in animals will work in man? And that's a huge gap. And a strength, however, the current field of cognition is that there's a focus on cognition from the point of view of Alzheimer's disease. Can we have better pills for Alzheimer's disease? Can we have new medications for schizophrenia, cognitive deficits in schizophrenia? There's autism and Down syndrome. So there's a big focus within pharma companies on cognition. So although there's a gap there, there's certainly considerable interest from pharma companies. We've heard about the pathophysiology of the transgenic mice of Down syndrome subjects. I'm going to take a different attack here, pardon the pun, and we're talking about looking at the pharmacology of a class of drugs we've known about for 50 years. These are the Benzodiazepines. So rather than look from the disease working upwards, I'm going to look at existing pharmacology and try and work backwards to cognition enhancers. So the first cognition enhancer was introduced again by Roche, a terrific breakthrough from Roche in 1960 with chloridase epoxide or Librium, followed shortly thereafter by diazepam, and shortly thereafter by any number of benzodiazepines. Also draw attention to Valium, as being made famous in the Rolling Stones' Mother's Little Helper. So we know that classical benzodiazepines like Valium do some pretty good things. They're anxiolytic, anticonvulsant, and myorelaxant. So those are good things. Also do things that are perhaps, depending on the clinical situation, are less good. So if you're taking a pill for anxiety, you don't really want to be sedated and at the steering wheel or operating heavy machinery. So this can be a bad thing, although benzodiazepines can also be used as hypnotics to put people to sleep. So whether these things are good or bad depends very much upon the clinical setting. Interact with ethanol, impair memory, induce tolerance and dependence. So all these things are collectively, depending on the clinical situation, could be viewed as bad. And I draw your attention to the fact that benzodiazepines do impair memory. So people that take benzodiazepines can often complain about feeling a bit fuzzy-headed. They can't remember things. They feel a bit detached. They do have cognitive deficits. And the benzodiazepines act on the GABAA receptor, and the GABAA receptor is a member of this family of receptors here. This is the cis loop receptor family, the CLR, which GABAA receptor is one member, nicotinic receptors is another. And the GABAA receptor is a pentamer, so it's 5 subunits. That's a generic GABAA receptor, and the GABAA receptor with benzodiazepine sites have an alpha 1, 2, 3 or 5, beta and a gamma 2. So that's the classical configuration of a benzodiazepine containing receptor. GABAA receptors or benzodiazepines combine a GABAA receptor and enhance the flux through the ion channel. So the ion channel is this hole in the middle. It's like a donut with a hole in the middle, and chloride goes into the cell, across the cell membrane. So chloride goes through this hole in the middle. You can have compounds which are inverse agonists, which, if you like, make the hole in the middle smaller, so less chloride goes through. You can have compounds that bind at this benzodiazepine site and don't affect the size of the hole in the middle, and they're called benzodiazepine site antagonists. And I'll talk about those a little bit later on. So this is just a cartoon to show how this is a GABAA receptor, so it's 5 subunits with a hole in the middle, and chloride flows through this hole in the middle from outside to inside the cell. So that's just with GABA there. With GABA and a benzodiazepine antagonist, it has no effect. So this compound binds very, very tightly to the receptor, but the receptor doesn't really know it's there. It has no effect on the function. With something like diazepam, so this is Valium, it kind of makes the hole in the middle bigger, so more chloride flows into the cell, and the membrane potential becomes hyperpolarized and the cell is less excitable. And an inverse agonist, as I mentioned before, makes the hole in the middle a bit smaller, so less chloride flows through. You have a depolarization and a more excitable cell. That's a very brief overview of what these kind of compounds do. So given that there's about 19 members of the GABAA receptor family, so in terms of the possible configuration of those 19 subunits into a pentamer, there's loads of them, hundreds of thousands of possible permutations. But actually, there's very few occur in vivo in the brain. And most of the GABAA receptors have a benzodiazepine binding site, so that's alpha 1, alpha 2, alpha 3 and alpha 5 containing receptors. That's about 3/4 of the GABAA receptor population, and there's some smaller populations as well. So most GABAA receptors in the brain have a benzodiazepine binding site, which I guess we already knew because benzodiazepines have quite a marked pharmacological effect. So again, just to restate the fact that these benzodiazepine agonists, like diazepam, increase the chloride flux and impair cognitive performance. Benzodiazepine inverse agonists do the opposite thing. They make the hole in the middle smaller, so less chloride flows through the GABAA receptor, and these enhance cognitive performance in animals. So the obvious question is, do they enhance cognition in man? So that experiment was tried in about 30 years ago, a compound called FG7142, and this is a report published in 1983, and this is a summary of that report published some years later. But what you can see is you've given this compound to normal volunteers, and the experience is hard to describe. It's existential, one subject reported, comparable to anxiety attacks, or possibly a pre-convulsive or a wave-like attacks of sheer, unfounded and extreme anxiety without any object. So you can see that these things are not the kind of stuff you would like to be having in humans. So that compound went no further, and that approach of non-selective inverse agonists went no further. But the question arose, is it possible to identify a subtype of the GABAA receptor associated with cognition? So if you're thinking of cognition, learning and memory, if there was one part of the brain you would identify as very important for learning and memory, it would be the hippocampus. And this is a rat brain, so this is the a sagittal section of a rat brain, so it's kind of going that way. That's the front of the rat brain, that's the back of the rat brain. And this is where the alpha-1, alpha-2, alpha-3 and alpha-5 subunits are found. We won't talk about the beta, gamma or delta, but the main thing is that alpha-1 is kind of scattered around the cerebellum and the cortex. And this alpha-1 subtype is the subtype that's targeted by zolpidem, or which is the sleep drug. So if you target this drug, people go to sleep. So that's Ambien. Ambien does that. But most importantly, the alpha-5 subtype occurs very highly localized within the hippocampus. So if you want to target any protein in terms of cognition, you'd like it to be highly concentrated in a part of the brain, like the hippocampus, associated with learning and memory. And this is a rhesus monkey brain, so we now have a coronal section, so that's kind of going that way. And we have here the cortex. This is all the benzodiazepine binding sites, all the cortex and the thalamus, the hippocampus. And with the alpha-5 subtype, again, we can see lights up the hippocampus. So this alpha-5 subtype is in the right place and implicates this subtype in learning and memory. Some additional mouse genetic and pharmacological data supports this hypothesis that alpha-5 is associated with cognition. I mentioned alpha-1 is sedation, alpha-2, 3 is anxiolysis. So all these data collectively give us two hypotheses. One is that an alpha-2, 3 selective agonist, which doesn't have any alpha-1 activity, will be a non-sedating anxiolytic. And that's a different story for a different day. I'll not talk about that. But the other hypothesis is that an alpha-5 selective inverse agonist, also called a negative allosteric modulator, should enhance cognition. So that was the hypothesis. So again, to restate this hypothesis, if a non-selective inverse agonist 7142 is anxiogenic in man, can we just target the cognition subtype, the alpha-5 subtype, without it being pro-convulsant or anxiogenic? So we don't want any of that nasty stuff that we saw previously with FG7142 in man. We just want the good stuff, which is the cognition enhancement. There's two ways of doing this now. I won't dwell too much on this, other than to say you can have compounds that bind much more highly affinity to the alpha-5 compared to alpha-1, 2 and 3. But when it does bind to any of these subtypes, it makes the hole in the middle equally small, so it has similar effects in terms of efficacy. Another approach is subtype selective efficacy, where a compound can bind to all four subtypes. It has no effect to alpha-1, alpha-2 and alpha-3, but has a big effect on alpha-5. And this is the approach that we chose, and I believe the Roche group have chosen a combination of this approach and this approach for their molecules. This is the compound alpha-5IA, alpha-5 inverse agonist, developed by Merck, and this is a publication by Jerry Dawson. You can see here it binds to all four subtypes, but has a big effect at the alpha-5 subtype and not much effect at the other subtypes. It's not pro-convulsant and not anxiogenic in rodents. And I thank Mara for explaining very nicely the water maze. And so this compound improves performance, and this is in normal rats, improves performance in normal rats in the Morris water maze. So that's a sign of cognition enhancement. So obviously the question is, well, what does this compound do in man? Well, it's reasonably well tolerated. We had a maximum tolerated dose of 4 milligrams in young subjects, and 4 milligrams was well tolerated in elderly subjects. We also did a study to show that the compound gets into the brain and binds to the receptors. So this is a PET study. We've heard about PET studies earlier this morning. So this is a subject that's given a placebo, so this is just a sugar pill. You then inject a radioactive tracer into the subject, and it binds to the GABAA receptor in the brain. And this is half an hour before placebo, half an hour after placebo, and 7.5 hours after placebo. And you can see that the binding of the radioactivity to the receptor is unaltered, which is exactly what you'd expect. With a test drug in 3 different subjects, this is the baseline scan half an hour before giving the pill. And then half an hour afterwards, you see there's less radioactivity bound to the brain. And that's because the drug has got into the brain, it sits on the receptor. so the radioactive stuff cannot bind to the receptor. So we can see here that this is significant occupancy of the GABAA receptor by this pill, and then some of that disappears as the compound is metabolized and cleared from the subject. So what we can say is that we don't get any anxiogenic effects in man, and that's at relatively high levels of occupancy. So it's not as though we don't have anxiety because the compound doesn't get into the brain. We know it gets into the brain and gives us high occupancy. So the question is, well, what does this stuff do? And we have very little data on what the compound does, other than a study from Dave Nutt's group. And this is a very nice little study. This is in normal volunteers. So you give them either the sugar pill or the compound. You give them 2 hours for their drug to get absorbed. Then you offer them half an hour of alcohol. And these are medical students, so it's very, very easy to recruit for this. You give them ethanol. I think this is about 6 vodka and oranges in about half an hour. You ask them to learn 20 words, and then half an hour later, you ask them to recall this. And these are the data. So these subjects are given placebo. So they give the sugar pill, then they have the vodka and orange, then they learn 20 words and have to recall it. And some of them are shocking. So a couple of people only remember 1 word out of 20. And performance is generally not very good. But if you're giving them the drug rather than the sugar pill, you can see that in this case, if they're pre-treated with the drug, they've gone from remembering 1 word to 5 or 6 or 7 words. So performance is improved. Clearly, this is not a patient population. These are medical students and relatively healthy, relatively normal volunteers. But at least this shows that the compound gets into the brain and does something. That compound got no further because that's the structure of the compound. It's soluble as brick dust, and any chemist would appreciate that water solubility is an attractive feature in a molecule. This is not very attractive. It gets metabolized by a hydroxyl group, which makes it even more insoluble. That caused renal toxicity, and that was the end of the compound. So another backup compound called MRK-016, similar to what we saw before, binds to all 4 subtypes and has most effect of the alpha-5 subtype. Single dose, 5 milligrams, well tolerated in young subjects. In elderly subjects, very poorly tolerated at 0.5 milligram. And that caused severe nausea and vomiting in 1 subject. Also, it had very variable pharmacokinetics. So this is, which is, when you give somebody a pill, you want to make sure that each individual you give a pill to has the same plasma level of drug. You can see here we have quite a lot of variability, which is not a very desirable feature in the drug. So this compound stopped due to poor tolerability and variable pharmacokinetics. So this Merck compound is stopped. Both compounds went into man, but it's important to say that both compounds were halted due to developability issues. Roche, as I mentioned, have a similar types of compounds in the clinic, so good luck to them. The alpha-5 selective compounds are clearly different from the non-selective compounds, and it's important to say that the hypothesis has not yet been tested because the Merck compounds progressed but had issues which are unrelated to the fact that they didn't work because they never got that far to test the hypothesis. So in terms of resources needed, I think it's clear that we need compounds suitable for testing. In terms of people, we need pharma companies that are willing to actually test drugs in Down syndrome, which is often not perceived as a very big market. Everyone wants to go for the big markets like Alzheimer's disease. Funding, well, whoever and wherever you can get that from, that would be desirable. And in terms of helping to create the resources, I think lobbying from patient care organisations and scientists themselves can greatly facilitate this. And obviously what we're trying to do is improve cognitive performance of Down patients and improving their quality of life. So with that, I thank you very much for your attention.
have any information on liver toxicity?
Yes, we do extensive preclinical studies in rat species and larger species, which is dog, and there was no sign of liver toxicity with either of those compounds. The toxicity we saw was renal toxicity with one compound, but the other compound showed no renal toxicity.
Just one question. We've also been interested in whether a drug for the brain might be improved in its pharmacokinetics, or at least in its toxicity, peripheral toxicity, by using a patch on the carotid to increase the concentration in the brain but not have a high concentration in the periphery. Has anyone tried that approach for drugs that would have counterindication in the periphery?
Not that I'm aware of. I mean, it's an interesting approach. I think in terms of pharmacokinetics and pharmacology, I think with the GABAA receptor, we're in good shape because it's primarily in the brain. So that helps. And I think some of the side effects we saw, like the tolerability, could well be related to the pharmacokinetics. So it kind of gets in the brain quickly, and your brain kind of goes, ooh, what's going on? And you get dizzy and you have poor tolerability. If we could have had the opportunity to flatten the kinetics and make it smoother, so you don't get that sudden rush into the brain, I think we would have got better tolerability. But
The other advantage is patch.
absolutely. So I think pharmacokinetics is really important with these kinds of drugs, because you don't want to hammer the brain with a sudden bolus of drug and your brain gets all kind of freaky and you feel poorly or whatever. I think pharmacokinetics is a really important aspect of these kinds of drugs.
I'm wondering, if just a more general question about how we get pharma interested in targets that we'd like to develop. Any general suggestions about how we get pharma hooked on Down syndrome?
And that's a very good question. In terms of things like Down syndrome, as I mentioned, pharma tends to be interested in the blockbuster, and the blockbuster is viewed as something that makes billions of dollars and a huge patient population. And we often talk about going for more segmented markets or smaller markets where you might have a better chance of showing efficacy rather than have a huge population of a whole spectrum of Alzheimer's subjects or schizophrenia subjects. If you go for a very defined population, you might actually have a better chance of showing efficacy. Now, we often talk about that, but actually doing that, we're still always attracted to the big blockbuster. So making a business case for smaller indications is difficult. And as for targets, what I think a refreshing approach for pharma at the moment is to say, there's loads of science goes on in the outside world. Let's accept that. We don't just do drug discovery inside. There's a whole lot of it going on outside in academic institutions. Let's start tapping into that. Let's start tapping into academic ideas, because there's some terrific stuff out there, and we need to reach out, because academics are often very willing to work with pharma because they have an idea and so expertise, and we have expertise in terms of the whole drug discovery process. So in terms of working together, it should be very mutually beneficial. I mean, there's a whole separate issue about who owns the intellectual property, but conceptually, the scientists in a drug company really want to work with external scientists.
It's an important theme, and I think this will come up at the roundtable, because I think there are new models that make this much more doable than the old models. Yeah. Okay. So, thank you very much.
Thank you.